Hi everyone and welcome to Chapter 6. Today we're going to focus on metabolism, including energy and enzymes. And anytime we mention metabolism, please make sure to remind yourself, well, what exactly does that mean? It means all of the chemical reactions in your body or in a cell. Okay, that includes building things up, breaking them down, and enzymes will help do that. Now, with your metabolism, we end up talking a lot about energy. And energy, the straight up definition, is the ability to do work. Now, there are two categories when it comes to energy. Energy can be kinetic or it can be potential. And so whenever you see kinetic, I want you to think energy involving motion, okay, movement. A lot of times we refer to that as mechanical energy. Okay, so kinetic is energy involving motion and it's mechanical, whereas potential energy is energy that's stored. It's the energy prior to motion, prior to movement. And a lot of times we call that chemical energy. Okay, so you notice in the images, whenever there's actual movement, such as the ball falling or you riding your bike up or down, that movement is kinetic energy, whereas potential energy is right before you have that nice burst of mechanical energy. So again, you could also think of them as kinetic is mechanical, potential is chemical. Now you also see this in nature, that constant flow between potential and kinetic energy. So for instance, you see solar or the sunlight as a potential energy, and it's a chemical energy within the plant, and then you end up having, once the animal feeds upon it, mechanical energy of the movement. So as he's getting that chemical energy turned into mechanical energy. Okay, so if you want, you can circle chemical and remind yourself that that is potential energy, whereas mechanical refers to kinetic energy, movement. Now, when we talk about energy, there's a few laws regarding energy. We call them laws of thermodynamics. Okay, they always use crazy big words to try and sound very, very smart with these laws. Now, the first law of thermodynamics is the conservation of energy, meaning that energy cannot be created or destroyed. It can only be converted from one form to another. And that's kind of like you saw in that previous slide, where you see that conversion of potential chemical energy into mechanical, physical, mechanical energy. Okay, at no point was the energy destroyed, and a lot of times you see energy converted into heat as part of reactions. So sometimes you think that you're losing energy, but it's not that you've lost the energy, it's that it's been converted to heat, which you see in a lot of the images below. Now, when we talk about examples of the first law of thermodynamics, the two that I have on this slide are photosynthesis and muscle contraction. So basically in these examples, you have some of the energy put in getting converted into chemicals, and then the excess energy gets converted into heat, which is a different form of energy, and that gets released into the environment. And notice that, it wasn't that energy was destroyed, it's just that energy got converted into heat that was then released to the environment, and then that heat ends up being a potential form of energy for other things within that system. The second law of thermodynamics, whenever you hear second law of thermodynamics, I want you to think of entropy. And there are two aspects of entropy. First off, energy cannot be changed from one form to another without the loss of usable energy. But again, notice that you're not losing energy itself. You're losing usable energy. So it's kind of like saying losing the mechanical kinetic energy, but It's that it's converted to stored or a different form of potential energy. Okay, so you're losing usable energy, but the energy is not getting destroyed. Now, the big part of entropy that I want you to circle, star, highlight, whenever people think of entropy, they focus on the aspects that waste energy goes toward increased disorder. Okay, so circle, star, highlight, increased disorder for entropy. And in the examples gave in the previous slide, so remember we talked about photosynthesis and muscle contraction as examples for first law. Those same examples can be used as the examples of second law of entropy, because in those examples you notice that some of the excess energy was released as heat energy. Now that heat energy that dissipates into the environment is no longer available to do work at that moment. It has to eventually be converted again. So even though that energy was not destroyed, it was not not usable in that exact form anymore. It became potential stored energy rather than the workable mechanical kinetic energy. And also when you think of heat being released, you can see that as increased disorder. Picture, molecules throughout that air just getting dispersed further and further apart, which is like disorder. Now, to visualize the cellular examples of entropy, focusing on the increased disorder aspect, when you have molecules getting broken down as part of metabolism, you see here, for instance, glucose getting broken down into carbon dioxide and water. Well, you notice that the single individual molecule is very organized, okay? But once it gets broken down, you have all of these smaller molecules, just like I mentioned a minute ago, dissipating out into the air, out into the environment. And that's increased disorder, okay? It's less organized when you see it, okay? You could kind of think of it as increased disorder. Some people think of it as chaos, okay? That's a more chaotic form. But it's more stable because that's how things naturally want to be. They want to, by that second law of thermodynamics, be more disordered rather than ordered. Okay. Now, you see that also in biology with channel movement. So here you have the hydrogen ions all in one place clustered together. And as we know, based on our discussion of plasma membrane transport, when you have a high concentration of ions on one side, it wants to balance out, right? So some of these hydrogen ions want to move across to the other side so that they're equal on both sides. But notice when you look at this, it's more chaotic, more dispersed everywhere, okay? It's less organized because rather than being clustered together, they want to be all spread out, and that's more disorder. Okay? If you have any questions or unclear what entropy means or are unclear of these examples, just send me a message in the Remind app and I will clarify it a little more. Now, moving on from those types of energy, we're going to focus on metabolism right now, metabolic reactions. Now, as I keep saying, whenever you see the word metabolism, I want you to think of all of the chemical reactions in a cell or a body. So that's building things up, breaking them down. All chemical reactions in a cell or a body. Now, when we say chemical reaction, there are two aspects of a reaction. There are the reactants and the products. The reactants are the substances that are participating in that reaction, okay? They are the things that are going to either get broken down or built up, depending on what type of reaction it is, okay? So the reactants are the substances participating in the reaction that will either be broken down or built up. The products are then the substances produced from that reaction. So for instance, if you've broken something down, well, now all of those smaller substances are the products. Now, we tend to look at these reactions and we ask ourselves, what's the free energy in the reaction, in the metabolic reaction? And that's the amount of energy available to do work. Now, we break that down into two different categories. I want you to know both terms, exergonic versus endergonic reactions, okay? Exergonic, remember previously I've said anytime you see X or exo in biology, it means outside, right? So exergonic are reactions that will release out energy, okay? Exergonic reactions release energy. This means that the products will have less free energy than the reactants because energy has been released. So they show that in the graph here. Notice the reactant energy is up here, the product energy is down here because energy was released. So there's less free energy now. Endergonic, well, end or endo is inside. So endergonic reactions absorb energy. They take in energy. So now instead the products will be up here because they absorbed or took in energy. They now have more free energy than the reactants. They have a greater ability to do work, okay, because they absorbed in. Now notice you can further break these two down to remind yourself that exergonic reactions, if they are releasing energy, it's because those products have been broken down. Digestion means broken down. Whereas endergonic, they've absorbed energy. These products have been built up. So remember, I said metabolism is both. You have some things being broken down, some being broken up. Okay, again, any questions, if you want more clarification, just send me a message. Now, when we keep talking about energy, as you already know, when we talk about energy in biology, we're talking about ATP. And remind yourself that the way that ATP is high energy or gives energy is by breaking that third phosphate bond. Okay, so ATP is high energy because it has those 3 phosphate bonds, and when you break that third phosphate bond, you end up releasing energy, which is usable by the cell because it's coupled to energy requiring reactions. Okay, so coupled reactions are reactions that will occur in the same place, same time, and the energy releasing reaction drives the energy requiring reaction, meaning that you have this allowance of ATP to drive forward energetically on favorable processes, okay, those energy requiring reactions. Okay, but again, make sure you know that the reason or the way that we get ATP from cells, energy from ATP is by breaking a phosphate bond that releases energy. Now, with metabolism, we said, it's all of these chemical reactions, it's building things up, it's breaking them down. And the way this actually happens in your body and in cells is through metabolic pathways. Rather than having one reactant, one product, there's usually a whole bunch of intermediates within that pathway. So to ultimately get to the desired end product, you usually have to have many different intermediate products. So for instance, A converting to B, to C, to D, to E, to F, before you get to the final one, G. And what's important is when you have this process, in order to help it along, the cell ends up using enzymes. Enzymes are biological catalysts. They are proteins that will help speed up a chemical reaction, but they are not used up or changed in that reaction. Okay, so keep in mind they help speed up a chemical reaction, but are not used up. They are not changed in that reaction. Now, what's important, as you see here, is with metabolic pathways, each of those intermediate steps usually has its own enzyme, okay? And you will not get that final G product if you don't have each of those individual enzymes present and functional. Okay, so make sure you're comfortable with the fact that metabolic pathways require multiple enzymes, one for each step of that pathway. Now, when we talk about enzymes, there's a lot of information we could discuss regarding how those enzymes actually work. How are they, speeding up that reaction? How are they helping that reaction? The first thing you have to know is that enzymes are binding sites for substrates, okay? And substrates are the fancy scientific way of saying reactants, okay? Each enzyme will have an active site, which is basically like in this picture here, that little, cut out area is the active site for the substrate or the reactant to then bind. Now, the way that that substrate binds to the enzyme can fit one of two models. And each of these models exists in biology in your body. It's just different enzymes have different models. The first one is the lock and key model. And the lock and key model is just like you see in this first little figure here. The active site of the enzyme will be a very specific shape to the substrate shape. And this active site shape will not change. Okay, so the lock and key model is that the enzyme will have a very specific active site shape matching the specific substrate shape, and it will not change. Induced fit, on the other hand, is like you can see here. It's very tough to see because it's very small change in that figure. But the induced fit model means that for some enzymes, the active site will sometimes undergo a slight change in shape to better fit the substrate. Okay, and that's usually the more accepted model, that the active site will change slightly to fit the substrate rather than already being perfect. Now, with that binding, we said that, these enzymes are helping speed up reactions. Well, how are they doing that? They're doing that by lowering what we call the activation energy of a reaction. Now, activation energy, if you think about it, activation, that means to get something started. So activation energy is the minimum energy required for a chemical reaction to occur. Okay, so the minimum energy required for a chemical reaction to occur. Make sure to write that down. So the enzymes are lowering that, but they are not changing the free energy, which is that energy difference that we discussed between reactants and products. Okay, now when you think of this, energy of activation is basically saying the energy required to initiate, to start up a reaction that, the reactants are a little sluggish and they need their push. Now, they're lowering the energy of activation, bringing those, substrates closer in contact with each other. Now, when we talk about lowering activation energy to speed up a reaction, the way that I like to kind of think of it or explain it to students, picture yourself saving up money for a new cell phone. And let's say right now that new beautiful smartphone is $1,000. Well, it might take you some time to save up that much money. That's a lot of money that you have to gather and save up before you can actually have that smartphone and start using it. Well, an enzyme, what they would do is lower the price. What if that smartphone is now only $100? Well, you're going to save up $100 a lot quicker than you would $1,000. So you'll be able to start using that smartphone a lot quicker, right? That's what we mean by lowering the activation energy, okay? They're making it easier or quicker for that reaction to start by making it so that it can more quickly reach that lower energy. And you see that in the figures on this slide. So in the first one, without the enzyme, you end up needing to get to that high of a peak of energy. Whereas with the enzyme, you only need to get to this low peak to start up the reaction. And it's a lot quicker and easier to get to that lower, cheaper peak if you're thinking in terms of the cell phones. Another way that I like to visualize the exact enzyme action, and you can retitle this if you want to write in your notes. This slide, what I'm saying now, explains the mechanism of how an enzyme works. And what enzymes do to help a reaction, I like to think of it like, a school bus seat. Picture first day of school or trip, students come into a school bus and they don't know each other, right? But on that school bus seat, two students who never met each other, they're now forced to sit next to each other. And what's going to happen along that bus ride? Well, because they're sitting next to each other, what do you do with someone sitting next to you if you're in a class? You start to chat with them, right? You start to interact with them and bond with them. And then by the end of that bus trip, well, what are those kids like when they get off that bus? Well, oh my God, we're best friends now, right? They've bonded, they've interacted because they were forced to be physically close proximity to each other. And that's what an enzyme is like. An enzyme is like that school bus seat. You have the substrates as the two students who end up sitting next to each other because the enzyme gives them an active site for them to sit close to each other. By being close to each other, then they can interact and bond. And then once they leave, they've been bonded. But what do you notice about that school bus seat, that enzyme? It was not changed. It was not destroyed. It was not used up in that reaction. It's still sitting there so that the next batch of students who come in, they can come in, sit together, bond, leave. Come in, sit together, bond, leave. Okay? And that's what enzymes do. That's their mechanism of action, is they provide an active site for two substrates, meaning two reactants, to sit close to each other so that they now are encouraged to interact and bond without the enzyme getting used up or changed. And that's, if you picture any reaction, if you're just, picturing a room, there are molecules all over around you that you can't see. It's hard for them to interact if they don't get close to each other, right? A molecule near your bed can't interact with a molecule near the outside of your house because they're too far apart. So the enzymes force things close together, close proximity, so they can interact. And again, make sure you write that down as the mechanism by which the enzymes are working. Now notice enzymes can help with either aspect of metabolism. They can help break things down or they can help build things up. Okay, so for breaking things down, you might have an enzyme where the active site encourages that substrate to sit there, and then once it's sitting on the enzyme, it has the time to degrade or break down into multiple products. Whereas synthesis is more like the two kids sitting on the school bus, where they bind to the active sites, they're close together, they bond, and now it's just one product. Now, because of the fact that enzymes are proteins, remember they're proteins, that means that there are a lot of factors that can affect how well these enzymes are going to work. There are a lot of factors that can make them more efficient, help more reactions occur, and there are a lot of factors that will slow them down or hinder their ability to work. One of the factors that can affect enzyme activity is substrate concentration, meaning how many reactants are present. If there are just a few substrates, as you see in this first picture, very few substrates, then there's going to be very few reactants, not a lot of enzyme activity. Because remember, molecules need to collide in order to react. Whereas if you increase the concentration of substrates, meaning increase reactants, boom, they're bouncing off the walls. They're more likely to hit into each other, more likely to have collisions, which in chemistry are reactions. Okay, so higher substrate will increase enzyme activity. They could have more reactions. There's also optimal pH and optimal temperature, and both are similar in the fact that they both relate to enzymes being proteins, and proteins have specific pH and temperature requirements. If you are outside of that pH or temperature range, you've just denatured that protein, meaning it's unfolded, it's no longer the proper shape, and it no longer will properly bind anything. It won't work right anymore. So if you do not have optimal pH and temperature, then enzyme function craps out. It decreases, it's reduced, and the enzyme ultimately can become inactive, meaning it's not working anymore. There's also things called cofactors and coenzymes, and both of these are substances that will activate an enzyme. They help enzymes work. So having cofactors and coenzymes present increase enzyme activity. They're required to help these enzymes work. The only difference between them is that cofactors are inorganic molecules, whereas coenzymes are organic. And the way that I remind myself which is which, whenever I see cofactor, I think factor, factory, and I picture a factory full of smoke and metal and bricks and all of this inorganic stuff. So cofactors are metals or minerals that help an enzyme work, whereas coenzymes are the opposite. They're organic, things like vitamins that help your enzymes work. And that's why making sure that you have a diet and supplements that are, a good amount of your minerals, of your vitamins are so important, because minerals and vitamins are helping your enzymes, which help all of your metabolic reactions happen. There's also enzyme inhibition, and we're going to go through each form of that one by one. So when it comes to enzyme inhibition, the first category is reversible enzyme inhibition, meaning any of the following can be reversed so that the enzyme will function again. Now, non-competitive inhibition, what that means is exactly like the name suggests. The inhibitor is not competing with the substrates or the reactants for that nice, comfy, active seat on the enzyme. Instead, what a non-competitor does is it binds somewhere else on the enzyme. Again, emphasize the fact it is not binding to the active site. Instead, a non-competitive inhibitor will bind somewhere else on the enzyme, and that triggers what we call an allosteric change. Meaning, so allosteric is this term that I'm highlighting over here. Allosteric change in that enzyme means that the shape of the active site has changed because of that inhibitor binding elsewhere. So now the substrate will come along and it'll no longer fit in that site. Okay, so you didn't block off the active site, but you were bound somewhere else and changed the shape of the active site. So basically the way you can picture this is if, let's say, you have a nice comfy seat in your office, right? Or your room. So you have that comfy seat, you fit perfectly in it. All of a sudden someone sneaks in and changes that seat to a children's chair. You try and sit down, you're not going to fit if it's a little children's sofa, right? So you can no longer fit there because they changed the shape of it. They didn't physically block you. They didn't cover that up. They just put something there that didn't fit. The alternative to that is competitive inhibition. What competitive is just like its name suggests. The inhibitor binds to the active site of the enzyme, meaning it physically blocks the active site so that the substrate cannot bind because the site is already filled. So an example of that would be like trying to park your car in a parking space that's already filled. If someone else parked in your space, they have physically blocked it. They didn't change the shape of that parking space. They didn't modify the space at all. They physically blocked it, and they were your competitor for that one spot. Okay? The last of the reversible enzyme inhibition examples is feedback inhibition. And as we mentioned earlier, when it comes to metabolism, you don't just have one reactant, one product, and one enzyme. Usually you have a long pathway, a full metabolic pathway with multiple enzymes along the way. What feedback inhibition means is a lot of times that end product, the very final product in that metabolic pathway, will turn out to be an inhibitor of one of the earlier enzymes. So for instance, in this example, the end product F is an inhibitor of the very first enzyme. And so it turns that off. It inactivates the first enzyme, so the pathway now stops. That kind of inhibition makes sense because think about it. If F is being produced in the body and you now have F, you have enough of that. Why would your body want to waste resources making all of these intermediates and making more of the end product when you already have it in your body to use? Okay, so just keep in mind that all three of these are reversible forms of inhibition, meaning they can be modified so that the enzymes no longer inhibit it. There are, however, some cases where inhibition of an enzyme is irreversible, meaning once that enzyme has been inactivated, you cannot rescue it. And what this means in terms of biology is there are materials that will irreversibly inhibit an enzyme, and we call them poisons. So when you hear poisons, they are irreversibly inactivating an enzyme. And some examples for humans are things like cyanide gas, heavy metals, things, like lead, nerve gas. Any of those are poisons that will destroy your enzymes irreversibly. So in this case here, in this image here, the heavy metal bound to an active enzyme. And by binding to that active enzyme, it changed the shape of the active site, similar to the way a non-competitor would be. But the difference here is that by binding, it bound in what we call a covalent form, meaning it is forever bonded to that inactive enzyme, and that active site is forever changed. Whereas in non-competitive inhibition, the competitor will temporarily, weakly bind to the enzyme, but can easily pop off. Okay, so poisons are irreversibles. In humans, I want you to remember cyanide gas, heavy metals, and nerve gas. So especially heavy metals, remember, since that's the example on the slide. Those are all poisons, irreversibly inhibit an enzyme. You can't fix it. And then in bacteria, a big example is penicillin. So when you take penicillin, the reason why it kills off bacteria is because that penicillin has a part of its structure that binds to the enzymes in bacteria that make the bacteria's cell wall structures. And that binding is irreversible. It's basically a covalent bond, just like in the example of humans and heavy metals. Okay, so once these poisons are bound to the enzyme's active site is changed forever, and you cannot reverse it. Something else we want to mention in terms of metabolism is that a lot of the reactions that we've been talking about, a lot of times we categorize reactions in metabolism as either oxidation or reduction. And you see it written as redox, so reduction and oxidation. a way to remember what exactly is going on is Leo says Gur. So that tells you lose electrons, oxidize, whereas gain electrons is reduction. Because in redox reactions, electrons are passed from one molecule to another. One of those molecules is losing electrons, and that is called oxidized. It has been oxidized, whereas the molecule that gains electrons has been reduced. Okay, and a way to remember that, if you think about it, gaining an electron, electrons are negatively charged, right? So they're gaining a negative, hence why they've been reduced. You've gained negatives. Now keep in mind that oxidation is taking place at the same time as reduction. So one of the molecules is losing electrons at the same time as one is gaining electrons. And the examples that you will see are photosynthesis and cellular respiration, both of which are going to have their own chapters dedicated to them. So as I mentioned before, with photosynthesis and cellular respiration, you're going to have chapters dedicated to each of those. But for now, I just want to briefly introduce you to them in the sense of what's being oxidized, what's being reduced, the fact that both of these processes, photosynthesis and cellular respiration, are examples of oxidation and reduction occurring at the same time. Now, again, keep in mind, when we talk about photosynthesis, that's how plants are making their food, making their energy. So in the photosynthesis reaction, plants are taking solar energy and using it to convert into chemical energy. And what they're doing is they take in the carbon dioxide that you breathe out and you water the plant. And with that carbon dioxide and water, they then produce glucose as a food source and the byproducts of water and oxygen. Now, in this reaction, when you're thinking about what's becoming reduced and what's becoming oxidized, remember, reduction is kind of weird when you think about it. I know when you think of something being reduced, you think of it having less. But in this chemical sense, it's being reduced because it has more negatives, right? So it's getting more negative, which is a charge less, right? So reduction is Gur, gaining electrons. And you notice that the carbon in carbon dioxide has gained electrons by having those extra hydrogens now with it. So the carbon of carbon dioxide has been reduced. Gur, it has gained electrons. Whereas the oxygen in water, Leo, has lost electrons, so it became oxidized, and you notice it no longer has the hydrogens with it. It lost those electrons. Now, when you're looking at these visually, a little trick that you can use to remember which is which is when something's reduced, you would expect it to get smaller, but in chemistry, it gets bigger. So if you remind yourself of that weird trick that the wording does with you, well then when you look at the example of cellular respiration, which is the exact opposite formula of photosynthesis, so cellular respiration is how you break down sugar to get energy. Well, what did we say? When something's reduced, it will actually look bigger. So notice that the O2 suddenly looks bigger with those extra hydrogens near it. Okay, so just looking at this formula, the one that got bigger is actually reduced and the one that got smaller has been oxidized. See how it's only 2 letters now instead of 3? But again, if you wanted to think of the science behind it, remind yourself, oxidized, Leo, lose electrons. It lost the electrons of hydrogen there. Gur, gain electrons, reduced. The oxygen now gained those hydrogens with it. Okay. If you're still uncertain or you have any questions, as always, make sure to send me a message and remind, and I will clarify further. Now, when we talk about this energy production that we keep mentioning, Energy production you have via the electron transport chain, and chloroplasts and mitochondria, meaning chloroplasts, the organelle for photosynthesis, and mitochondria, the organelle for cellular respiration, both generate ATP via electron transport chains. And what electron transport chains are these membrane-bound carrier proteins. So again, you've seen carrier proteins in membranes in the previous chapters, but these guys, instead of passing, the ions that we've seen in other slides, they're passing electrons from one carrier to another. And what they do is they start with high energy electrons and low ADP. So here they're representing the electron transport chain as if it's a staircase, and notice it starts with high energy electrons. And at that point there's low ADP. Every time an electron is passed to the next carrier, which they show as bouncing down a step, so this would be high energy electron passed to this first carrier, what happens, the cell then captures and uses that energy to make ATP, okay? And each time the cell, each time you're getting that high energy electron passed to the next carrier, the cell is capturing more of that energy and using that energy to make ATP. So as it's going down, the cell keeps capturing more and more energy. So by the end, it's now low energy electrons. And there's high ATP because the cell has gathered and collected and synthesized a lot of ATP as the electrons were passed carrier to carrier to carrier. Okay, so you started out with high energy electrons, low ADP, and at the end, it transferred each step. Each step is a different carrier protein along the electron transport chain, and now you end up with low energy electron at the bottom, and the cell has gained a lot of ATP for synthesize that has been synthesized. Now the way that the cell is actually synthesizing that ATP throughout that electron transport chain is with something called chemiosmosis. Okay, so make sure you remind yourself that whenever you see the term chemiosmosis, that means ATP synthesis that has been coupled to the electron transport chain. And basically what this is that in addition to the electrons, there are certain carriers of the electron transport chain that will be pumping hydrogen ions. And those then build up on one side of the membrane, okay? Because the cell is pumping all of those hydrogen ions across that carrier, there's now a buildup of hydrogen ions on one side of the membrane. This big concentration of hydrogen ions on one side of the membrane now causes an electrochemical gradient, and that provides energy for ATP production, okay? And then there are special channels within the plasma membrane. So within the membrane, this carrier protein right here, this channel, that's called ATP synthase, okay? Synthase because it can also be considered an enzyme structure. And so this ATP synthase complex produces ATP as the hydrogen ions flow through it. Okay, so as the hydrogen ions collect in this location, they then flow through the ATP synthase complex, which produces ATP. And that production is called chemiosmosis. Now, you don't have to, look into the crazy biochemistry or organic chemistry pathways of how this production is occurring. Just be aware of the general terminology. So be aware that chemiosmosis uses ATP synthase complexes to pass hydrogen ions through a gradient that then produces ATP. okay, because ATP synthase is the enzyme producing or synthesizing ATP, okay? Again, if you have any questions or uncertainties, or if you want me to recommend any videos that might help or map things out in a different way, just let me know. In the meantime, we have now finished another chapter, and this is an important chapter to finish because Chapter 6 is the end of week 1. That means you now have everything you need to make sure you complete your study guide and submit it ASAP, and to now make sure that you head on over to the discussion board if you haven't done so already, and make a post with 5 multiple choice questions in it. You do not give the answers to your own questions. You just write a single post in which you put 5 multiple choice questions that can be from any of the chapters of week 1. And then you go to another student's post, a post that has not been replied to by anyone yet, and you put what you think the answers would be to their 5 questions. And you can either just put the letters, or you can actually explain the reasoning behind your answers. And if you think that someone might have made a mistake in answering any of the questions that you see on the discussion board, then you can let me know and I can address it and explain the correct answers. Or you can very politely, always be very polite to each other on the discussion board, comment with what you might think the correct answer is and why. And you could always refer to particular slides. Okay? Make sure you upload the study guide for the week directly into Blackboard. There will be a place for you to put it in the week 1 folder. So make sure to do that. Make sure it is not in a PDF. It has to be in a Word document so that Turnitin can scan it for plagiarism so you can get full credit. And make sure to head over to the discussion board. Okay? As always, contact me with any questions or concerns, and have a wonderful day.